Hi, I'm Larry Johnson. I am a professor at Texas A&M University, and today we're going to talk about Introduction to Microscopy 2, types of light microscopy, and immunostaining of specific structures.
And there's different, we'll mostly look at stains tissue with bright microscopy, but there's other types of microscopy that will allow you to see live cells. So with Toluidine, we can see these columnar type cells, simple columnar cells that's in the intestines. We can see these rods, which are mitochondria. You can see a thick structure in through there, which is a striated brush border. There's a brush border through there. We can see junctions here at electron microscopy that we see just as terminal bars in through there. So by using electron microscopy, we can supplement the detail that we can see with light microscopy. Also, epithelium is sperm is another one. We can see the nucleus of a sperm, we can see the tail, and we can see the axoneme with little dining arms that are located in the sperm tail. Also, with microscopy, you can use immunofluorescence, namely that you can label something with a fluorescent tag, and if it binds to that, then you're able to visualize the fluorescent tag. So it's like you can put the cowboys in a group, let them all have white hats, and then you can identify the cowboys by the white hats is some of the methods that we have. And here you see I have a fluorescent probe add to an antibody, hence immunofluorescence, and it binds to an antigen or a structure that it wants to identify. And so here you can see where the structure is identified now by this fluorescence. And you can do that with peroxidase, or you can have gold particles, have different size gold particles. And this is important for electron microscopy. These two are important for light microscopy. Auto radiography, so if you bake a cake and you use a radioactive sugar, then the cake will be radioactive. And the same with cells. If you feed the cell something that goes in a certain structure as a precursor and it's radioactive, it will be radioactive in that structure. And here we see some secretory granules that were fed radioactive amino acids, and indeed those amino acids show up in the vesicle that's associated with producing the protein. Carbon replica is another way of visualizing cells. This is typical transmission EM. Carbon replica, you're actually just looking at the replica. You're not looking at the tissue itself. And that is where you blew in carbon in through there, and carbon will fill up on one side, not the other side, so it gives you different shades of it. Like here we can hardly see the nuclear pores, but there you can see nuclear pores very nicely in the carbon replica.
We want to thank the many original sources of images, figures, drawings that may have been modified for this presentation. We want to thank those in this slide. That's the end of Introduction to Microscopy 2, types of light microscopy, and immunostaining for specific structures. Thank you.